All right, so now as we begin looking more closely at cellular reproduction, I'm, I'm just going to give you guys a little bit of an overview of what the cell cycle is. So, this is going to be a cycle where the cell actually spends a lot of its time either arrested in development, synthesizing DNA, or preparing to divide. So this is the overall cell cycle, and we always start kind of right up here with G1. So the cell is going to go through G1, and it can actually stay arrested in that state, and which we call G0. And then once it's going to have to go through a checkpoint, which we call the G1S checkpoint. Saying that the cell is complete and is ready to go forward with DNA synthesis. Once it passes that checkpoint, it's going to go into the S phase, and the S phase is where DNA is going to duplicate or copy itself. And once in the S phase, it proceeds into G2, where the cell is going to prepare for mitosis. After the G2 cycle, it's going to come up against the G2M checkpoint. And this is where the internal mechanisms are actually going to check and make sure that that cell has synthesized its DNA correctly and is able to divide. Once it passes the GM or G2M checkpoint, then that cell can proceed into mitosis, as well as cytokinesis. And this is what we really call the M phase. So the cell, let me back up. The cell is actually going to spend a great portion of the time in interphase, which is what we really call the first part of the cell reproduction process is interphase, and it's going to spend a very small portion of the time in the M phase. So here's just a few features of the cell cycle. Just gives you the stage over here on the left-hand column, and then in the center gives you the major features. So this is a really good table for you guys to study from if you're making flashcards, so that you can put the different stages on a different card and kind of lay them out in the cell cycle. That'll give you a really good idea for each part of the cell cycle, its structure, and its function. So the cell cycle ends with the M phase, and the M phase can either stand for meiosis or mitosis. We're going to concentrate mostly on mitosis in this part of the lecture, and then we'll get into differences a little bit later with meiosis. So the number of the chromosomes as well as the DNA molecule is going to change during the cell cycle and the mitotic process. And so you really have to have a good idea of how many chromosomes should be present to identify what part of the cell cycle that the cell is actually in. So to determine the number of chromosomes, it's easiest to count the centromeres. So that's why we spent so much time on part B of this lecture learning about the centromeres. If you want to count the number of DNA molecules, so also known as the copies of DNA, then you count the chromatids. And those chromatids are going to be the individual chromosomes. So it would be like chromosome 1 from mom. That's one chromatid, whereas one chromosome from dad would be another chromatid. So let's start looking more in depth now at the mitotic process. Again, we start with interphase, which is the longest part of the cell cycle. In interphase, we can see that we have our cell here, and it has a plasma membrane or a cell wall along the outside. That's that dark yellow line. And in the center is going to be the nuclear envelope. And within that nuclear envelope is going to be that DNA material. It looks pretty swirly. It's kind of hard to see. Okay, so the centrosomes are going to be just kind of very relaxed within the nucleus at this point in time. All right, so then we're going to proceed into prophase. So this means that the cell has passed that G2M checkpoint and is ready to divide. The first part of the M phase for mitosis is prophase. In prophase is where the chromosomes are going to condense. They're going to actually shrink down into those traditional X shapes that we're used to. and the mitotic spindle is going to form. So that mitotic spindle is going to be in the form of a centrosome, and it's going to start alienating itself to either side or either pole of the cell. We then proceed into prometaphase. And prometaphase is where the nuclear membrane begins to disintegrate. So we had a full nuclear membrane here in prophase. However, in prometaphase, that nuclear membrane starts to disintegrate because you have to be able to divide the cell. That leads us into metaphase. And metaphase is where the chromosomes are going to come together and randomly line up on the metaphyseal plate. Okay, so this is one point of genetic variation in meiosis, and we'll talk about that later. So, but for in mitosis, just realize that those chromosomes are going to come together and line up on the metaphyseal plate. From metaphase, we proceed into anaphase, and this is where we see separation of the sister chromatids, and those chromatids are going to begin moving towards the opposite walls. And finally, in mitosis, we end with telophase or telophase. And telophase is where the chromosomes are going to arrive at the spindles or at the poles, and the nuclear membrane is then going to reform. Cytokinesis is going to take place, and we're going to get two, not necessarily new cells, but two cells where we only had one in the past. So looking at a different configuration of this process, again, we have interphase where our nuclear envelope is going to contain DNA in its relaxed form. We're going to proceed to prophase where our spindle fibers are going to be migrating towards opposite poles. That leads us into prometaphase. That nuclear membrane is going to go away. And then in metaphase, those chromosomes are going to line up on the metaphyseal plate, and those sister chromatids are going to separate from one another in anaphase. And finally, in telophase, we have cytokinesis taking place where that new cell is going to form, and more specifically, the cell wall is going to form between the new sets of DNA. All right, so that whole process of the sister chromatids splitting away from each other and migrating towards the poles is actually a really interesting concept. So each chromosome each sister chromatid is going to be attached to a spindle fiber. And that spindle fiber is going to be made up of microtubules. And these tubules are what is really going to attach to the sister chromatid. And the microtubule is made up of a protein called tubulin. And tubulin is going to be like a two-part protein like we see here. And it's going to have lots and lots of different subunits that all come together to make up this microtubule. So each microtubule also has a polarity to it. So one end of that tubulin subunit is going to be positive, one end is going to be negative. And this is really what drives the whole process of taking a sister chromatid from the center of the newly forming cell and taking it to the pole. So chromosomes are going to be pulled towards the poles by kinetochores. And this is done via a molecular motor. So as you can see here, this gives you just a little bit of an overview. We have our chromosomes here. We have sister chromatid number 1 right here. It's going to have a kinetochore associated with it that's going to have a positive charge on one end and a negatively charged second end that is going to be more towards the pole. And that microtubule is going to be made up of tubulin subunits. So how do chromosomes migrate to the opposite poles? This is going to happen during anaphase. And the first part of this process is that tubulin is going to depolymerize as the chromosome is pulled towards the pole. So again, we have our sister chromatid right here. We have a kinetochore where the spindle fiber is actually attached, and that spindle fiber is then going to start moving this chromosome towards this pole. If we look more closely at what's going on here, the tubulin is starting to depolymerize, which means that we're going to see movement from the positive side to the negative side because opposite charges always attract one another. As those charges are attracting one another, we are going to be losing tubulin subunits. Okay, so as the motor proteins move along the chromosome, the tubulin subunits are going to be removed at the end so that the strand actually gets shorter and shorter and shorter as we get towards the centrosome. All right, so we will end there with this section and pick up with sexual reproduction and genetic variation.